Why does a treatment that cures cancer in one patient not work in another? Cancer is a frightening diagnosis, but these days the prospects of a successful outcome are better than ever. The DNA in each of our cells gets damaged up to 1 million times per day, but we have elaborate proofreading mechanisms that quickly find and repair the damage. Sometimes a mistake does get through, but usually the change does not cause any harm. However, occasionally one of these variants is not harmless and knocks out one of the repair genes or activates a gene that makes cells divide faster. One variant by itself is not usually enough to cause cancer, but over time another mutation may occur, and then another. Eventually, some of these cells begin to divide more aggressively and ignore commands from other cells to stop dividing, forming a tumor. If the tumor is found before it gets too big, sometimes it can be removed with an operation. Even if it cannot be removed completely, we can also treat cancer by targeting rapidly dividing cells using radiation or chemotherapy. But these methods also affect normal cells that divide quickly, such as hair follicles and epithelial cells lining the digestive tract, which is why some cancer patients experience hair loss and nausea. Actually, there is another way to treat cancer. If we know exactly what went wrong in the DNA to cause the cancer, then there's a chance that we can find a drug designed to counteract it. There's a problem, though. Most tumors have dozens of variants, but only a few of them actually drive the cancer, and only some of these might respond to treatment. When a patient does not get better with the standard treatments, the doctor submits a request for a comprehensive genome profiling, or CGP, to explore treatment options with the hospital's genome expert panel. The job of the expert panel is to help match cancer patients with cutting-edge therapies based on the specific mutations in their DNA. First, a pathologist prepares a sample of the tumor and sends it to a company like Foundation One or GenMineTop for sequencing. A machine like this NovaSeq 6000 breaks the DNA into short fragments and copies each one millions of times using special nucleotides that glow in the dark with a different color for each base. The machine takes a picture each time a nucleotide is added and writes down A, C, G, or T depending on the color. The machine produces huge files containing the DNA sequence for each read. Then the company searches the human genome to figure out where each read belongs and assembles them like the pieces in an enormous puzzle. When the reads are carefully lined up, some columns show a checkerboard pattern where the reads disagree about which nucleotide is present. These are variants or mutations, and the company sends us a report of the most important ones. A bioinformatician analyzes each variant to identify the most dangerous mutations that likely cause the most harm. Genetic counselors examine the patient's family history to see if cancer runs in their family. A diverse team of oncologists and researchers search the literature for any treatments that might be effective against that variant. Many new drugs are being developed, but it takes a long time, and often the only way to get access to cutting-edge treatments is to enroll in a clinical trial. Pharmacists search our custom database to try to help the patient find a suitable clinical trial. We call it HopeDB because it offers one last hope even when all other options are spent, and the patient asks, "Isn't there anything else I can do?" The Cancer Genome Expert Panel is an interdisciplinary team that includes volunteer members with a range of skills from almost every department in the hospital. Cancer is scary, but it is encouraging to know that patients are in good hands at Hiroshima University Hospital.